Good afternoon, everyone. My name is Karine Yahya, and I'm presenting my honors project for the genetics class, and I'm talking about DNA repair, and the Nobel Prize for this research was awarded to Thomas Lindhall, Paul Modrich, and Aziz Sanjar. I'm going to start this presentation by talking about Tom Lindhall, and he's a Sweden man born in January 28th, 1938. He studied at Karolinska Institute in Stockholm, where he received his doctorate and became a professor after in Princeton University in New Jersey and Rockefeller University in New York. He developed an understanding of how living cell works and the cause of cancer and aging processes. He worked at Clare Hall Laboratories as a cancer research, and he was affiliated with Francis Crick Institute and Clare Hall Laboratories at the time of the award. He started his first DNA research with his professor Eleanor Hammarsten, the behavior of DNA in glycol, and he received several awards like the French Prix Étranger of INSERM in 2009, the Royal Medal of the Royal Society in 2010, and the Copley Medal, which is the highest medal in the Royal Society, in 2013. Now, Paul Modrich, he's an American man. He's an American biochemist who was born on June 13, 1946 in New Mexico in the United States, and he received his doctorate at Stanford University in 1973 and Massachusetts Institute of Technology. He works at Duke in North Carolina, and he did his postdoc with Charles Richardson at Harvard Medical School, and he showed how methyl signals repairs incorrect replication of DNA. He was affiliated with Howard Hughes Medical Institute and Duke University School of Medicine at the time of the award. Now, talking about Aziz Sanjar, he's a Turkish geneticist. He was born on September 8th, 1946. He studied at both Istanbul University and University of Dallas, Texas, and he became a geneticist when his background, where his parents have no education. At the time of the award, he was affiliated with UNC Chapel Hill. He was appointed as the chief medical officer in Turkey. He did his medical practices in a very small room in his family's house for apparently 6 months, and he was paid by the Ministry of Health, and he did won a NATO fellowship to fund his PhD research. He chose the US because it's a leader in scientific research in the world. He was admitted into Johns Hopkins University in 1971. So Thomas, Paul, and Aziz got the Nobel Prize for mapping and explaining how cell repair its DNA and safeguard the genetic information. They worked at the molecular level. So let's begin with talking about the base extinction repair by Thomas Lindhall. This research came to Lindhall's mind when he started to heat the RNA. It's known that the RNA is more sensitive than the DNA and degrades rapidly. So Lindhall thought if RNA was destroyed quickly, what will happen to the DNA? Could the DNA molecule be stable for a lifetime? So in the past, it was believed that DNA is stable, but Thomas Lindhall demonstrated that there is a lot of chemical stabilities that makes DNA unstable, like UV radiation, free radical, carcinogenic substances. So he did his research in Sweden, in Karolinska Institute in Stockholm, to open the door on a new research, where he concluded that there must be a molecular system for repairing all those DNA defects. There must be something that counteract the amount of DNA damages. So he discovered that there is a bacterial enzyme that removes remains of cytosine from DNA. So he published The Instability and Decay of the Primary Structure of DNA to discuss DNA damaging and repairing process in 1993. He started the Cells Toolbox for DNA Repair to observe the BER, the base extinction repair, by removing the small non-helix distorting base lesions he's repairing, or it's repairing the damage. So as we can see in this picture, cytosine can easily lose an amino group, so it forms a base called uracil. But as we know, that uracil cannot pair with guanine. So there's an enzyme, so that's a DNA damage, because guanine and uracil do not pair. So an enzyme called glycolase discovered the defect and excised the base of uracil. So after that, there's two of the major DNA specific exonuclease. According to the Science China Life Sciences, they're called TREX1 and FEN1. TREX1 is a 3' to 5' exonuclease, and FEN1 is a 5' to 3' exonuclease that remove the rest of the nucleotide from the DNA strands, and they're the DNA polymerase that fill in the gap, and the DNA strand is sealed by DNA ligase. However, even if the DNA is stable, as we said, it can undergo changes, and even if it's in the protective system, it can be damaged by UV radiation. So repairing the DNA damaged by the UV radiation is developed by Aziz Sanjar, the nucleotide extinction repair. So before that, he did his research where he succeeded in cloning photolyase, the genetic enzyme that repairs the UV damaged DNA. After his research has been shelved, he continued working on the DNA repair. He found out that the bacteria has two systems for repairing UV damage. The one with photolyase, as we can see in this picture, and the second one, there's a system found out that works in the dark. So for better understanding this system, Aziz and his colleagues used 3 UV sensitive strains of bacteria that carried 3 different genetics mutations: UVRA, UVRB, and UVRC. As we can see in this picture, they worked with these strains, and they tried to analyze them. After isolating and characterizing the enzymes coded by those genes and grounding in vitro experiments, these enzymes can identify the UV damage and making 2 incisions in the DNA strand, one on each side of the damaged part. So as we can see in this picture, the UV radiation can make 2 thymines bind to each other incorrectly, which is a damaged DNA. So the enzyme exonuclease finds the damaged and cuts the DNA strand. So those enzymes that are from those bacteria that he did analyzed do find the damage and cuts the DNA strands. So a fragment of 12 to 13 nucleotide is removed, and then the DNA polymerase fill in the resulting gap, and the DNA ligase seeds the DNA strand. Now the injury has been dealt with. And Aziz also discovers that human and bacteria has same UV damage repair. They function similarly in all organs in the body, in the human body. After Aziz Sanjar, Paul Modrich has examined a series of enzymes that affect DNA during his postdoc in Harvard. Those enzymes are DNA ligase, DNA polymerase, and the restriction enzyme E. coli. Toward 1970, DAM methylase, which is one of the enzymes, has shifted his attention. So he thought, or he have a thought that DAM methylase couples methyl groups to DNA. Those methyl groups could function as signposts and help a particular restriction enzyme to cut the DNA and at the correct location. However, the molecular biologist Matthew Meselson did not agree with Modrich. He started his experiment where he constructed a bacterial virus with mismatched base pair. So he mismatched the base, A with C, T with G, that do not pair. And he was surprised that it was fixed. Those damage were fixed by itself. Meselson thought the methyl group helped repairing the DNA. This is where Meselson and Modrich ideas crossed. Meselson and Modrich worked together and placed the DAM methylase that Modrich was working on And the mismatched DNA, the virus with mismatched DNA that Messelson was working on, and the bacteria corrected the DNA lacking in the methyl group. So they found out that the DNA mismatch repair is a natural process. As we can see in this picture, there's two enzymes that called MUTS and MUTL that detect the mismatch in the DNA. The enzyme MUTH recognize methyl groups on DNA. Only the original strand, which acted as a template during the copying process, will have the methyl groups attached to it. The faulty copy is cut, the mismatch is removed, and the DNA polymerase fill in the gap, and DNA ligase seal the DNA strand. He then, Modrich, was able to recreate the complex molecular repair mechanism in vitro, and he published it in 1989. So we get exposed to sun, cigarettes, or genotoxic substances, so our DNA gets altered, and for each cell division, mismatch repair, correct thousands of mismatched. Our genome would fall apart without these repair mechanisms. The fail of one of those mechanisms may increase the cancer risk. The damage of the nucleotide excision repair process will cause xeroderma pigmentosum. So people who suffer from this disease will develop skin cancer if exposed to UV radiation. As we can see in this picture, how this girl is affected by the xeroderma pigmentosum, and this is one of the failure of the nucleotide excision repair. And there's also another cancer, which is the colon cancer, and it's a defect in the DNA mismatched repair, and it can increase the risk of developing hereditary colon cancer. So this research explained how cancer cell DNA is unstable because their repair system is switched off. This is why cancer cells mutate and become resistant to chemotherapy. Those cells are dependent on the repair system. Without it, they'll be damaged. So inhibiting the repair system will allow the damage of the cancer cell and stopping them to grow. So since those cancerous cells are dependent on the repair system, so those repair system, if they were damaged, they will inhibit the growth of those cancerous cells. So one of the pharmaceutical that was invented is olaparib. Olaparib is one of the pharmaceutical, and it's a PARP inhibitor. PARP means poly-ADP ribose polymerase inhibitor, and which is the enzyme that activate those cancerous cell. So it's inhibitor to this enzyme. So by blocking the PARP, the cell becomes overwhelmed and will cause the breakage of the double strand, leading to the cell death. So by inhibiting this enzyme, it will cause to the cell death. And as we can see in this picture, there's something called BRCA mutated cell. The BRCA are the tumor suppressor. So when those are mutated, those mutated cells, so when PARP is inhibited, BRCA become the mutated cell. The tumor suppressors are now mutated, and they can cause the breast cancer. So those mutated cells, they have no repair, no homologous recombination, and it will cause cell death. And then now another pharmaceutical or another medicine that have been discovered is the berzosertib. The berzosertib is another pharmaceutical that inhibit the ATR protein, which respond to the damaged DNA. Some type of cancer rely on the ATR protein to making those repairs. By inhibiting the activity of ATR, berzosertib interferes with the ability of rapidly dividing cell to defect damaged DNA. So as we can see in this picture, when this ATR is damaged, when this ATR is inhibited, there's cell death. But when this ATR is not inhibited, as we can see, that there's DNA damage repair survival. So inhibiting this ATR will cause cell death and will decrease the number of cancerous cells. So this Nobel Prize has developed our knowledge about the base repairing system and how this would help for cancer treatments also. Not only for how to learn about the base repairing system, but also to create some treatments for the cancer. And Paul Modrich said, that's why curiosity-based research is so important. You never know where it's going to lead. A little luck helps too.